Hi, this is Miss Litton, and this is my wonderful Period 1 AP Bio class. Say hi.
Hi.
And we are discussing Chapter 11, Mendelian Patterns of Inheritance, and we just discussed the idea of genotype versus phenotype as part of the introduction. Genotype is what is in your DNA, and phenotype is how it is played out. And I just posed the question about meiosis, and I talked about the law of segregation, and I asked you the question, we know that when things get segregated or separated, what stage of meiosis are we thinking of overall? Anaphase. And we know in meiosis there are two, and there's anaphase 1 and anaphase 2. And then I asked you to think about which one of those is most critical when we're talking about the traits that we inherit. So I said make a claim and provide evidence. So anybody want to make a claim about meiosis 1 or meiosis 2, anaphase 1 or anaphase 2, and in our inheritance, the traits we receive? Anybody guesses? Scared? What gets separated in anaphase 1? Homologous pairs. What are your homologous pairs? They are the same size, same shape, and they what? Code for the same characteristics, and one half of the pair came from your mother and one half of the pair came from your father. Okay? So that's what's getting separated in anaphase 1. In anaphase 2, what are you separating? Sister chromatids. And the only reason there would be variation in your sister chromatids would be due to what? Crossing over. And when does crossing over occur? Prophase 1, right? Otherwise, they would be similar. The only changes you would have is due to crossing over. However, the homologous pair, could they be quite different? Yes, because one you inherited from your mother and one you inherited from your father. So when you do anaphase 1, you are separating, you have no idea how those homologous pairs could line up. That's called the law of independent assortment. They can line up any which way, and you could either get your mother's or your father's chromosomes, and this is increasing variety. So that would probably be a very key event, that anaphase 1, in determining what traits you receive. If you think about it, your mother could be carrying within her a trait from her father that's bad and a trait from her mother that's good. And you hopefully are going to get the ones that is what? Good. But it's a 50-50 shot which one she's going to give you. Do you agree with that? And then you combine that with whatever your father gives you, then that is going to be your genotype. And then it's going to express itself in its phenotype. And we understand chromosomes, but Mendel did not. He is known as the father of genetics. And throughout his lifetime, he did research primarily with peas. I'll talk a little bit more about him in a minute. But it wasn't until about 50 years after his death that the significance of his work was understood. And had Darwin had a chance to look at it, that would have helped him so much, because he would have understood, had evidence for where variety could have come for natural selection and evolution. And Mendel died without his work ever being recognized for what it was. So let's get a little bit of background on him if you go in your notes. He was an Austrian monk who studied math and science at the University of Vienna, and so that's why I gave you a picture of it. And he was a substitute teacher, and he had farmers for parents. I'm telling you this because what did I tell you he worked with? Peas. So he had an understanding of farming, okay? His work went unrecognized during his and Darwin's lifetime. And before this, okay, there was this idea of blending, this blending concept, that you are a mix of both of your parents. And there are some traits that play out that way. It's called incomplete dominance, when you cross a red flower with a white flower and you get a pink flower. There are some things where it appears like blending, but due to Mendel, there was a particulate, think like particulates, and those are chromosomes, and we know them as alleles, and you get 2 copies of each allele for the trait that you will have, and this accounts for the differences. So on your Mendel's particulate, before Mendel blending, blue, tell them A. And And B, it did not help Darwin because it did not account for variations. Did not account for variations. Because if all we did was blend, 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 we'd just have a great big amalgam, right? No differences. All right, so Mendel's particulate theory of inheritance, traits are inherited as particles. Offspring receive a particle from each parent. Now we would call these particles genes. And so these are his P's, and he looked at several different traits, and he used statistics. He used math to analyze his results. He prepared his experiments carefully and conducted a lot of preliminary studies. And the reason why he worked with the garden pea, Slate, tell Blue, why did he work with the garden pea? Okay, and he chose 22 true breeding varieties. So he looked at 22 different traits. Now, by true breeding, okay, he would take, for instance, comparing tall pea plants with short pea plants. Tall pea plants are dominant over short pea plants. And it's not like you became a medium plant when you bred a tall with a short. What happens when you bred a tall with a short, because tall was dominant over short, if it was a true breeding tall plant, that means both of his alleles, both of the alleles said tall and tall. So all they had to give was what? Tall in their gametes. Now when I say all they had to give, in your mind you should be thinking through meiosis 1 and meiosis 2, right? So if they have a homologous pair, be right here with me. Be right here with me. Both say tall. What's gonna when are the DNA gonna replicate? As stage of interphase, right? Now all of the sister chromatids are gonna say what on them? Tall. There's no option for anything else. These are true breeding tall plants. So after you could do synapsis, you could do crossing over. It doesn't matter. What are you going to get every time for height? Tall. So anaphase 1, you're separating the homologous pairs. Anaphase 2, you're separating the sister chromatids. Every single one of these is going to say what? Tall. That's all they have to offer. Okay? You cross that with a true breeding short plant. Here's the deal. In order to express the short trait, both of your alleles have to say short. Because if just one of them says tall, you're going to be tall. That's the law of dominance. Okay? So 3 laws I've already introduced, and we haven't even got partway through the lecture, and these are important. The law of dominance would be something like tall is dominant over short. The law of segregation says you're going to do anaphase 1, right? And you're going to separate mother's chromosomes from father's chromosomes. The parental chromosomes are going to get separated, and you're getting one or the other. You can't get both. Right? You can't get both. And then the law of independent assortment is saying, remember metaphase 1, the way they line up? That they can line up any which way they want, so you could get any combination of parental chromosomes. Okay, so those are the 3 laws we're going to hit. So he chose 22 true breeding traits that were very simple, and he kept careful records. He had a lab notebook that he wrote everything in, yes? They actually went through his notebooks. And he was very, very detailed. I'm going to say he had a little bit of OCD going for him, for sure. I mean, he had such careful records. And from that, he developed these 3 laws that are still used today. Are there exceptions to those rules? Yes. Okay. So he compared things like tall versus short plants, round versus wrinkled pea seeds, and also yellow versus green peas. And most of the peas we eat are what color? Green. Actually, the dominant color is yellow. And then this is where I would tell you something else. That just because a trait is dominant does not mean it's most prevalent. How many fingers do you have? 5. That's a recessive trait. The dominant trait is 6 fingers. My name is Mantigo Montoyo. It kills my father. Okay? Now, 6 fingers is dominant. Has anybody had a friend who has 6 fingers? Your band teacher was born with 6 fingers, and I have pictures, baby pictures of him with his 6 fingers. Yes, that is a dominant trait, and he had it surgically removed. He has no problem me telling you that.
Why would he have it removed?
It's not like functional, like I can play the piano even better. Boom. Okay? It's not that helpful. Yeah. And I have that picture. I need to look that picture. But you can, yeah, you can Google it. Yeah. 6 fingers is a dominant trait.
6-finger baby.
Yeah, 6-fingered baby.
Like a really cool band name.
6-fingered baby.
It is. 6-fingered baby. All right.
Hella nasty.
Now,
Where do they cut off the 6 fingers?
okay, guys, come back to me. When you would cross a tall pea plant versus a short pea plant, a true breeding tall pea plant versus a true breeding short pea plant, I'm going to jump ahead just because I know you can handle it. Okay? All right? Do you know what this is? A Punnett square. Okay? So don't worry about this right here. If I am crossing a true breeding tall pea plant, so this pea plant is very, very tall. Oh, I want it to be green. Very, very tall. Okay? True breeding. He is big T, big T. Both of his alleles on his chromosomes, okay, have that dominant trait. Okay? And you cross that with a short pea plant. Now this is what you do. With recessive traits, you don't put S for short. You put a little what? A lowercase t. Okay? So if it is a short pea plant, how would we, if I looked at its homologous pairs, what would its letters be? Little t, little t. Okay? So when you cross them, and what you typically do is you would take the 2 alleles, and you just say, okay, so anaphase 1 is going to separate them. All of these will have the same version of the T, and all of these will have the same version of the T, right? So I only need to address what are the 2 that I have, and both of them are big T. And all they have to give is what? Little t. Okay? So these are your gametes. Let's say this is the egg, and let's say this is the sperm. Okay? So what happens if this sperm right here, if this sperm fertilizes this egg, then you're going to get big T and a little t. That is referred to as the genotype, the actual alleles that they inherit. Phenotype is what it looks like. What will it look like? This individual will look what? Tall. Perfect. And you know what? That's going to repeat itself for all of them. And that's what it did for Mendel, is he would cross these true breeding tall with true breeding short, and all the offspring would be tall. It doesn't mean that there's 4 offspring, right? There could be hundreds of offspring, but I'm just separating out the homologous pairs, okay? And then these are the inside of the box. These are the potential zygotes, the potential offspring. Yes?
So those are all the potential, but the actual plant will only have one of those boxes as like phenotype? Or genotypes?
No, every offspring that it has, it depends on what sperm. You're thinking about humans, so we would only have 1, and these are the potential, right?
Sure.
Yeah, but yes. But every time they mated them, we're talking like in a population, every time they would all turn out tall.
But you could do the same test for like people. Right?
Absolutely, we're going to get there.
Okay.
Yeah, we're going to get there. Okay, so now these are the parental generation. This is the P generation right here. This is referred to as the F1 generation. So these are like the parents, and these are the kids. Now we're going to mate the kids together. It's not gross because they're plants. Okay? So now the F1s are going to have sex with each other. They're going to reproduce with one another. And now it's not the kids, but the grandkids, right? So let's try that. So now we're going to cross a big T little t with a big T little t. Now, I think it would be very helpful for you, and I know we never use paper in here, okay? But to get out a piece of paper and a pen and title this piece of paper, Chapter 11, Practice Problems. Now, if somebody wants digital documentation of what they're doing right now, and you want like a digital document, get an iPad and get a pencil. If you want that. Yeah.
I would do it.
You have to get the one that matches your iPad.
Oh, okay.
So maybe get your iPad first and then get a pencil.
Wait, I have 523. Somebody use 523. This is way cooler. And paper. Paper sucks. Oh, this is
All right. So I jumped way ahead in the notes, but I felt like you were ready for it. So are you surviving? Okay, and then we'll go back and fill in the notes. If your pencil needs to be charged, okay? If your pencil needs to be charged, can I demonstrate? We've never, these have been your option to use all year long, okay? If you take your pencil and you just take the cap off, the cap will stick magnetically to the iPad. So just put it on there so you don't lose it. And then you just put this in the charging station off the end. Just like where you would, go ahead, show 'em.
Oh.
You see how that works? All right.
I know, right?
Okay, so now come back to me. So now we're crossing an individual, okay, who's big T, little t with big T, little t. Okay, those are our two individuals. So this individual, due to the law of segregation, can either give a big T, look at me, a big T or a what? Little t. So I'm going to put those right here, big T or a little t. And the second individual can either give a what? Big T or a little t. Now this is what I want you to do. Okay, I'll model it in one box for you and then you can repeat. Okay, look to me. So on the top, I want you to go, okay, if this sperm right here fertilizes this egg, the genotype, okay, what the alleles say, is going to be big T, big T. The phenotype, how is this individual going to look? How is it going to appear to us if they're big T, big T? Tall. Okay, so in your boxes, give both your genotypes and your phenotypes. By convention, you write the capital letter first before you write the lowercase. When you are done writing it out, give a double tap to your partner's desk, and then you can, when you're both done, you can compare. But try to do it by yourself first. I only skipped over 15 slides.
Miss Litton,
Yes, baby.
can you find the genotype for like more than two like offspring or more than two parents? Because like on the Bozeman's, he was trying to find like 4 of them, and then he wanted to find like which one would be the recessive genome or like parents or something.
I'll talk to you about that once we get going a little bit more. Okay, now, anybody need a little more time on here? Okay, so check your work with me. So what will the genotype be here? Okay, do it with me so I know you know it. What is it?
Big T.
Big T, little t, and what's the phenotype? Tall. And what's the genotype? Phenotype? Tall. Genotype? Little t, phenotype? Short. Muy bueno. Now, look, I know as much Spanish as Trump. Okay, so how many different genotypes? Sorry, hashtag nasty woman. I just don't have a pantsuit. How many different genotypes do we have up here on the board? How many different genotypes do we have on the board? 3? Okay, you're right, there are 3. Let's write them from most dominant to least dominant with the ratios. So watch me to make sure you get it right. Okay, so what's the most dominant genotype we have up there? Big T, big T. So my genotype ratio is 1 big T, big T. What's going to go next? 2 what?
Big T,
Little t, and 1 little t, little t. What is my how many different phenotypes do I have up here? 2. What's my ratio? Yes, and don't make it naked. Say 3 tall to 1 short. Okay, now, check with your bio buddy and say, bio buddy, how you feeling about this? Are you doing okay? Go ahead, have that discussion.
I learned this in
All right, so let me just back up a little bit. So when you look at a chart like this, okay, what he did is he would make these crosses between true breeding traits, and he would cross the dominant, a true breeding like purple with a true breeding white, and then all of them would turn up purple. All the F1s would turn up purple. So he would say purple is dominant over what? White. But when he crossed the F1s together, just like we did in our last cross, what trait started popping up again? The white. Okay? So it wasn't gone, because if it was blended, they would just be lavender, right? Instead of tall and short, it would just be medium. So you're crossing this, look at me, a tall trait with a short trait, and then all the offspring are tall. So you think, what trait is gone? You think shortness was gone, right? But then when you cross them, shortness popped up again. So shortness must have been masked, but it was still there. Okay? If I have brown hair in real life, could I have a blonde-haired child? Yes. The very fact that I have normally auburn-colored hair, that's a recessive trait. So I am masking a recessive trait. And if my husband had blonde hair or reddish blonde hair, then can you see how we have the chances of having a blonde-haired child, of which I do. I have a brown-haired child and I have a blonde-haired child. Okay? So that's the same thing here. So take a look here. Oh, here. Come on, it's a little funny. There, have you look. Menu, pea soup. And those are his experiments. Well, it's a little funny. All right. So he would do these reciprocal crosses. So he could take one's pollen and fertilize the egg, and take the other one's egg and with the pollen, so they could have each of it. That he knew it wasn't just a factor of the egg or just a factor of the pollen, because he would do these reciprocal crosses. I would like Blue to explain this picture working from left to right. Go ahead.
the plant makes children and has peas. And then the grandchildren get smooth
All right, so a couple things I want to point out to you. It didn't matter whether the pollen came from a wrinkled plant or the pollen came from a smooth plant. It still was inherited in the same way, because he did those reciprocal crosses to check that. So all of them turned out to be round. The trait is round versus wrinkled. But then when he crossed the F1s again, the wrinkled trait came up in about a 3-to-1 ratio. And then, whose turn is it now? Slate, go. You should have no problem with this. Go ahead. And carry it over, Slate. All right. Okay, did you fill in all your Mendel and garden peas part? So we're at Mendel's laws. the law of dominance, Mendel's law of dominance says that one trait or factor can be dominant over another. He called them factors. We know them as alleles. Okay? So, for example, tall plants are dominant over short plants. Youngest bio buddy, use this chart for the law of dominance to talk about the law of segregation. Use this page to talk about the law of segregation. How does that come into play? Go ahead. I gave you the law, I think.
It's the alleles have to be the dominant, or
Okay, so the law of segregation, we could talk about any of them, but you see it more in this one right here. The law of segregation says, let's say you're going to make pollen from this. The pollen's either going to get the big P or the little p. That's the law of segregation. Okay, so right here, youngest bio buddy, use this. Explain about the law of segregation. Go ahead.
Yeah, the homologous pairs separate from teeth either doing anaphase or don't know what else to say, really.
Now, Mendel did all of this without knowing anything about meiosis. There was no microscopy at this point. He couldn't see the cell. He figured all of this out just from his careful records and his breeding, this particulate theory of inheritance. Okay? Never, ever, ever saw meiosis in his lifetime. Now we can look at it and go, oh, okay, now that makes sense, right? Yes?
Are there only two kinds of genes, like one dominant, one recessive? Like
No, I'm just opening the book a little.
Okay.
I'm just opening the crack in the door here. We've got more to go. Okay, so when we look here, okay, the law of segregation says for this individual, he can either give the big G or the little g, and this individual can either give the big G or the little g, right? That's your law of segregation. All right, let's write some notes about that. In summary, each individual has two factors, which we now call alleles, for each trait. The factors segregate. What's another word for segregate? Separate during the formation of gametes. Do you remember, what did we say during meiosis? What can we call those cells when we're done with meiosis 1? They are no longer, yes, they are no longer diploid. They are what? Haploid. Why? Because we separated the homologous pairs. Okay. Each gamete contains only one factor from each of the pair of factors. And then fertilization gives each new individual two factors on homologous pairs for each trait. Right? This is a homologous pair of chromosomes. Let's say the red one came from mom and the blue one came from dad. What is going to happen during the S stage of interphase?
replicates.
Yes, they will replicate. Right? And this one will replicate. The original red and blue are still the homologous pairs, but now we've duplicated, right? And so we're going to see that same sequence is going to be exactly the same, right, on our original versus our replicate, right? And the only thing that's going to break up this, notice these are all dominant, big G, big R, big S, and then this one is a little t. The only thing that's going to break up this linkage that we have right here is what? Crossing over. Okay? All right. So on your notes, an allele is defined as alternate forms of a gene. So here, alleles at a gene locus, right here. The G's are right here on that chromosome every single time. That's where the G's hang out. Alternate forms of a gene that occur at the same gene locus, a specific location on homologous chromosomes. a dominant allele will mask a recessive allele. There are exceptions. There are exceptions. Okay, when you look at the genotype, you can refer to the genotype as homozygous or a homozygote or a heterozygote. What does hetero mean? Different. Homo means same. So if it's a homozygote, it's like big T, or what? Little t. Both alleles are the same. Perfect. And then I gave you a heterozygote, right? The phenotype is the appearance of the genotype. The appearance of the genotype. All right, time to assess ourselves. Okay, so the dominant trait is a widow's peak, and the recessive trait is a straight hairline. Yes?
I have a question. If you had like two parents with two like recessive traits, like say like you had a parent with like blue eyes and hazel eyes, which one would like?
Hazel is a modification still in the recessive end of things. Now, I'm going to tell you there's a lab. Did I not talk to you about this lab that I do? There's a lab I do in Honors Bio where we look at a series of our traits, and there are dominant and there are recessive traits, and if your parents are around or you can visit, then you record your parents' traits as well. And I had a student, he sat right there at the lab bench. It was a crowded class, and he was I loved him. He was a loud student, like you knew he was in the room, but he meant well. He was a good boy. He wasn't trying to be, bless his little heart. And there's a series of traits that you look at, and the very first one was like, he had already gone, I have him go home and look at their parents, and his parents had blue eyes, both his parents, and he had brown eyes. Now, blue eyes are recessive over brown eyes, okay? So, in order to have blue eyes, you have to get a blue from each parent. You have to be blue, right? Like, I have blue eyes, my husband had blue eyes, so the only eyes we had to give our children was blue, okay? So, I can still remember him sitting, he goes, Miss Sun, but my parents both have blue eyes and I have brown eyes. I'm like, they're modifier genes, right? When you look at your eyes, it's not like it's a solid blue. There's like flecks of like yellow and orange, right? And your eyes don't change color, okay? Your clothes change color, and those pull out different colors. So please don't say that to me, okay? Now, when you're first born, a lot of babies are born with blue eyes, and then their pigment sets in later. That can happen. Or you can be born with blonde hair, and your pigment sets in, and you're really going to have brown hair, or vice versa. That's different. That's maturity. Just as I'm getting older, my hair turns gray, right? That means I'm all, oh, there's modifier genes. Literally, we get to the next trait. And he's all, Miss Sutton. And again, he had a dominant trait where both his parents had the recessive trait. And then it was like, Miss Sutton. It was, my little heart was like, and now it's like I'm very careful when people, so when you first asked me that, I'm like, that's why you can see kind of like, what are you going to ask me? But time to have the talk, okay? So it was like a series of traits. And there are some things, that can still be modified, and we'll talk about that, exceptions to the rule where it's not straight up. But hazel and green are kind of modifiers on the blue. When you see somebody who has blue eyes, it's really the black of their retina reflecting through because they have no pigment on the front of their eyes. It's lack of pigment. And that's why I always say it's sunnier outside for blue-eyed people. And they can have more damage to their retina because they don't have any pigment to protect the front of their eyes. And my blue-eyed people unite, raise your hand. Okay, we are mutants. Okay, that is a mutation. It's not necessarily adaptive to have blue eyes, right? Because we can have more damage to our retina. Okay, yay, mutants. All right, so now let's take a look at your hairline. So widow's peak is dominant. Recessive is a straight hairline. Have your bio buddy help you and tell me, are you A, widow's peak, or B, straight hairline? Let's go, you guys. It shouldn't take that long.
Good guy, Frank. Jake, you have to punch it in.
Sean. Oh, Sean. You missed the fan.
Here you go, peeps. Let's go.
Just not get 100.
We're not gonna get 100.
Okay, and then I need like 3 volunteers.
Wait, what is
3
it?
volunteers.
Jeremy.
1, 2, 3, 4. Come on up. Sterilize your hands. You're gonna distribute. And put this in a clean place. Don't put them on the desk because the desks are gross. Okay? And these are expensive. That's why I'm just kind of being careful with it. You're going to get one of these. Don't do anything with it. Just put it somewhere clean. Okay? We're going to need this in just a minute.
Gosh, that's gross. SPA. You messed up.
Yeah. Okay, and then guys, give me any extras back in this too. Make sure everybody gets one.
I'll start with this one.
Okay, guys, while they're doing that, pay attention to me. Okay?
Not 100.
Ah, okay, now, take a look up here, okay? What trait was most prevalent in the room? Straight hairline. Is that a dominant or a recessive trait? Recessive. So this shows you that just because a trait is dominant does not mean it's the most what? Prevalent, okay? So, yeah, just put the extras back in there. We'll need them in just a minute. All right, so now take a look here. In order for you to have a widow's peak, okay, you can be big W will give you a widow's peak right here, okay? Big W, little w will still give you a widow's peak, right? And the only way to get a straight hairline is what? Little w, okay? Do you know who all the people are?
Fox,
Vince Vaughn.
Bird, Bear,
All right.
Cowboy Bear.
Okay. So, genotype, I want to refer, like you're referring to these alleles right here. And then phenotype, you would have to look at the individual. So, for instance, if I wanted to have normal wings, how are all the ways I could have a normal wing? If normal wings are dominant. Big N what?
Big pig.
Okay, so okay, so big N would be homozygote, right? And that would give me normal wings if I was big N, right? If I am big N, little n, that's a what? Heterozygote, but that would still give me normal wings. The only way to get vestigial wings is if I am what? Little n, little n. Yes?
Does the order matter for heterozygote?
No. Okay? Because it would, you know, you're on either side, right? It doesn't matter. It's just your 2 copies that you have, one from each parent. Okay? All right, so now, in your book, you have practice problems. And I am going to strongly recommend that you do those practice problems. And I'm actually not 100% sure that that's the right page because that's the old version instead of the new version. Okay, but when you get in your book, there are a series of practice problems and do them. Okay, I'm not going to do them with you right now, but I want you to do them. And then we can talk, if you do them, we'll talk about them in class. Okay, but I want to give you a chance to practice them. Okay, but what I do want to do is talk about this PTC paper. Okay, not PCP, PTC. Now, for some of you to taste this PTC paper, okay, is a dominant trait. To not taste it is a recessive trait. You will know when you taste it, okay? But before we taste this paper, okay, you're going to do 2 things. First, I want you to rip a little corner off of some piece of paper, regular paper. And I want you to chew on it because you need to, that's your control.
chew.
Yeah, just regular paper you pull out from your notebook. Good job. Just a little piece of regular paper. I just want you to try, because it's, okay, you chewing on it? Normal paper. Just normal paper. And the reason why I want you to chew on normal paper is because it's not like normal paper is that yummy, right? So I don't want you to think, oh, I can taste it, right? You need to have the idea of what paper tastes like. Okay, now, your little strip that I gave you, I want you to rip it in half. That little strip of PTC paper, rip it in half. You're going to save half because you might want to test somebody else. Now, this is what I want you to do with that paper. On the count of 3, we're going to put it on our tongues and rub it to the roof of your mouth. Okay, everybody holding their half? 1, 2, 3. Rub it in your mouth.
Oh, that's you.
Okay.
Oh wait, my goodness.
You can spit it out when you're ready to spit it out. All right. Rinse your mouth, chew on, pull something out of your lunch if you want to. Yeah. Well, you're not identical. Now, guys, you're going to give me yes or no. If you tasted it, you know you tasted it. It's like spraying perfume in your mouth, okay? But worse. If it just tasted like, it's kind of like paper, no big deal, then no, you didn't taste it. So click in yes or no, could you taste it? I don't know. If you're like, I don't know if I tasted it, then you didn't taste it.
But I wasn't freaking out.
I know it's hideous, right? I'm a taster. You can see I did not taste it. I've done it enough times.
I knew it was gonna taste her. I didn't taste it, but like I knew it was gonna taste her. I was like, she's playing with me.
All right. Now, in this case, the dominant trait was most what? Prevalent. If 2 tasters have a child, will they always have a tasting child? If somebody is a heterozygote, right? If 2 parents are heterozygotes for tasting, what are the odds they would have a non-tasting child? 25%. And there is some debate that because they are taste receptors, that if you are a heterozygote, you might taste it a little bit. Like, how many of you in here say, well, it's not hideous, but it was definitely different than paper. There was something going on, okay? You are probably heterozygotes for it, truly, okay? So let's answer this question. Fun with biology.
A bad left side.
I kind of wish I tasted it.
Hey guys, remember your tasting because we're going to be doing a lab later that refers back to this. And if you don't want to, so remember you probably will remember.
I want to taste it now.
Let's go, annoying Facebook girl.
Okay, yeah.
Nail polish remover, because you've tasted that a lot. Oh, we did not get 100. Tasting is a phenotype, right? Tasting is a phenotype. It is not your genotype, okay? Tasting, tall, short, round, wrinkled, that's how you display it. If I just say tasting, because there's different ways to be a taster, right? I could be big T taster, right? Or I could be what? Big T, little t. Yes?
If I could taste it very strongly, does that mean both my parents are big T, or is that?
No, not necessarily. They just happen to both give you what?
Dominant.
Yeah. I would expect both your parents, right? If you think about this, if it's a dominant trait and she can taste, then her parents could be homozygous dominant or heterozygotes, but they should still be able to taste, right? They may not taste it as strongly as she does if they're a heterozygote for it. So that's why I said save half if you want to have your parents, check your parents. Yes?
How does it work? Like the strips?
What?
The strips?
Oh, they dip it in a chemical that some people can taste and some people can't. Its sole purpose is to do genetic studies in
Like
classrooms.
GMOs.
Yes.
Is this like the same thing where like some people think cilantro tastes like soap
Yeah.
and then others think it tastes
More,
like
yeah, that would be a full genetic thing. Cilantro.
Do you have any quesadillas?
Now, you guys, now there's a difference here. There's preference, right, and appreciation for something, right? Or there's, oh my gosh, this tastes hideous. Like, I hate olives. Some people prefer the taste of olives. I think they taste like dirt. Okay? But I had a cousin who wanted them in her Christmas stocking, and she would put them on each of her fingers and like eat them off her fingers, and I'm throwing up in my mouth looking at it, right? So I love Brussels sprouts now, but as a child I did not prefer them. But I love to cut Brussels sprouts in half, toss them in a little olive oil with garlic salt, and roast them. They are delicious. Now, who would disagree with me?
Jake.
Okay.
Yeah.
See?
I'm the only one.
Now, who would agree with me? That Brussels sprouts are delicious. Delicious.
As long as you don't burn them.
Yeah.
I like the little crispy edges. They're like potato chips.
I hate the crispy.
Okay.
I like that sauce.
How dare you? What is it?
I don't like cherry soda, but some of you do.
Oh, cherries like Dr. Pepper spicy cherry soda.
I don't like soda at all.
That's what it is defined as. Dr. Pepper is defined as a spicy cherry soda. Doctor, alright, ready? I'll look it up.
Let's go, let's go, let's go. Commit. Commit.
Max.
Stop. Look at me. Look at me. Have you submitted your answers yet? We are waiting. Okay. I'm waiting. Show your clicker to your bio buddy right now. Okay, I'm done.
Hey. Ooh, look at that.
Last college student.
It's lazy college student.
Whatever. Yeah.
Last college student. Last
Yeah,
one on the
exactly.
planet.
Yeah.
I felt like
I'm from New
I
Zealand.
wasn't
Yeah.
Cilantro.
Stop mocking her. Max. Yes.
Is it possible for someone to
I can't hear.
Is it possible for someone to be all dominant traits and all recessive traits? Or like,
Probably statistically that would be very difficult. Could it happen? Yeah. Is it likely? No. Let's go, peeps. You're too slow. I don't know what to tell you. Let's go. This is varsity. See if I can get it. No.
Oh, look who's number one.
Still waiting.
That was the last one.
Oh.
Who's number 2? Who's number 4? Yo, Mondays all around.
Okay. So hairlines, we already talked about it. What's dominant? What I want you to do is cross 2 heterozygotes for widow's peak. Give me the geno and the phenotypes. Go. Quickly. Make a Punnett square. Cross 2 heterozygotes for widow's peak. I want you to do it all by your onesie on that. Remember I said keep a little scratch paper out? Or do it on your iPad. Do it all by your onesie. Don't look at anybody else's until you get the double tap. If you're struggling in your setup, I'm giving you some hints right here.
You better be ready. It does.
You know, yeah, funny genes recessive manager. Funny genes recessive.
If they're both, then that means that they're different. So that means there'd be a big one and a little one and a big one.
I guess the funny genes
Because I think what you did all big ones. Or I don't know.
Okay, did you do it? Okay. Is that what you got? Oh, whoa.
Yeah, I did.
All right. Easy peasy?
Lemon squeezy.
Lemon squeezy? All right.
Let's go.
Clarifying questions, anyone? All right, let's go. Try this one right here. Not out loud. Hear me when I speak to you. Hear me. You may get this and it's like, oh my gosh, this is so easy, I can't even stand it. And that's good, okay? But your bio buddy may not be at the same place as you. Do not make them feel afraid to talk to you about how you're working it out. So go, really? You know when somebody goes, you go, how are you doing? And they're like, fine. And you're like, really? Okay. So make sure they're okay.
Should we give the ratios?
Yes, always give me ratios.
Yeah. Oh no, I did not taste it. No. I think like that. Tried it. I'm all about it.
Got your genome phenotype ratios? Did you double tap? Where can you find more of these practice problems? Yes. And then where do you think the answers are going to be? In the back of the book. Do you think you could Google genetic practice problems and find more? Yes, of course you could. Do you think we're going to do some null hypothesis and some chi-square tests on the results?
Yeah.
Oh yeah, we are. Okay, is this what you got? Did you get that? Now, would you write your ratio 2 to 2? No, what would you write? 1 to 1. Good. Write it 1 to 1. Always simplify your ratios. See if your bio buddy simplified. See if they got the same answer. All right. Now, can we please level up? All right. Questions? Concerns? All right. So now, you can, whoa. Try that. Okay. All right. Gotcha. Okay. We're going to talk about the product law. Okay. If I was going to flip 2 coins, okay, what are the odds that I will get heads with this coin? Okay. So I had a half of a chance that I would get heads and a what? Half of a chance that I would get tails. Do you agree with that? Okay. And then would you say that's the same here?
Yes.
Okay. So a half a chance heads and a half a chance tails, right? Okay. So we got a heads tails combo. I want to do an easier one for you to start off. Let's see if it works. There, perfect. Okay. So we got a tails combo. If I was flipping 2 coins, right, what are the odds that I would get tails, 2 tails?
The order.
What? Doesn't matter the order.
Okay, it's a combination.
Okay, so how do I do that? This one had a chance of getting tails 1/2 of the time, right? And this one had a chance of getting tails 1/2 of the time. And the product law is you multiply each of their probabilities together. So what's 1/2 times 1/2? 4th. So together I had 1/4 of a chance of both coins turning up tails. Do you see a Punnett square?
Oh, yes.
If I had 2 heterozygotes, big T, little t, big T, little t, what were the odds of getting little t, little t? 1/4 of the time. So you're going to be like Punnett squaring in your head, right? Because we're only looking at 1 trait right now, but we're going to level up and look at 2 and 3 and 4 traits. Those are some ginormous Punnett squares. So we're going to be moving into the product law in order to figure that out. So I had 1/4 of a chance of getting tails, tails. What were the odds that I would get heads, heads? 1/4 of a chance of getting 2 coins that were heads. Do you agree with me on that? Now, here's the part where we're going to look at some other opportunities. Look at your notes real quick. Go to Mendel and the laws of probability. And I skipped over independent assortment. I'll come back to that. The product rule of probability, okay, is where I multiply together what are the odds of each incident happening. Okay, so I had 1/2 of a chance of this and 1/2 of a chance of this. Now I'm at 1/4 where for each. What about a heads tail combo?
100%.
Okay, well, heads tails. So heads and tails. What were the odds of heads tails? 1/4. But I could have also gotten a tails heads. What were the odds of that? 1/4. So it doesn't matter about the order, right? So I could have gone heads tails or what? Tails heads. So this is when you add them together. What is 1/4 times 1/4?
Okay.
Right? If I add 1/4 and 1/4, I get a 1/2. I had 1/2 of a chance of getting any kind of heads tails combo. Do you see that? Okay, so we're going to use that to kind of level up a little bit here. So again, there are practice problems I'm going to want you to look at. Okay. Okay. Let's do this one. Let's do this one right here. Freckled parents have non-freckled children. What is everyone's genotype? Freckled parents have non-freckled children. What is everyone's genotype? Discuss with BioBuddy. Okay, do the parents have freckles?
Yes.
So what's their genotype? What? So how would I write that?
Big T, little F.
Capital F, lowercase f, and dad? And none of these children have freckles. What is their genotype? Little f, little f, little f, little f, and little f, little f. Okay? What you need to remember is chance. When we talk about chance, it's not going to go, oh, you had a tall plant, and a tall plant, so you need to have a what? Short plant. Every single event, you look at the same probability. There's 1 in 4 chance of them having a freckless child, and they did. And the next child they had a 1 in 4 chance of having a freckless child, and they did. Okay, so you look at it individually, at each individual event. All right. I know, trying to see if I, oh, it's right on here, sorry. So on Mendel's laws of probability, I just went over independent assortment. I'm going to come back to that. Law of Mendel and the laws of probability, the product rule of probability, we did that. 1/2 times 1/2 times 1/4. And then the sum or additive law of probability. Additive. Yeah. And then number 3, chance has no memory. Each offspring has the same probability of inheriting a trait. It is not dependent on the genetics of the offspring before it. Chance has no memory. Each offspring has the same probability of inheriting a trait. It is not dependent on the genetics of the offspring before it. All right? Now, I want to talk to you about a test cross. Let's say we have a purple plant and we cross it with a white plant. Now, I know white plant, that color of flower, is recessive. But when I have a purple plant, I don't know if it's purple, right? If it's homozygous purple or it's heterozygous purple, because either genotype will make it purple, yes? Who do I want to cross it with to find out its purpleness? A white one. Why do I want to, youngest bio buddy, explain, why would I want to cross it with a white one? And what would the results tell me? What would I expect? Go ahead. All right. So if I cross it with a white plant and all the offspring turn out purple, I know it's homozygous purple. I can determine its genotype, yes? That's if I have a lot of crosses, right? Not just once and done, but a lot of crosses, okay? And if just one plant turns out white, I know it is heterozygous for being purple, yes? Okay? And so that's what's referred to as a test cross. So for instance, if we have a brown-haired child, we have a brown-eyed person, not child, that's even worse. Who would we need to find out if we want to know if she's brown or she's carrying the blonde gene? Who would we need to cross her with? What?
Where are we testing hair?
Hair. Yeah, we would need to test cross her with Jeremy. Okay? And then we could find out. Let's say they had one child and that one child had brown hair. Does that mean she's brown? No, we would need to have several children with Jeremy, right? And then if they have 10 children together and all of them, they're very productive. If they had 10 children and all of them turned out to have brown hair, then we could probably safely assume that she is what? Brown. But if they just have one blonde-haired child, we know that she is what? Brown.
Is there a better way of testing that, or do they have to just like make?
No, I'm just giving you an example.
Okay.
All right. First world problems, let's go. Yay! 3. Come on now.
I know, I heard.
Let's go. I want to ask you one more question.
Why do you go kiki?
This could tie your, what? Okay, so if just one, if all their offspring were tasters, then the unknown parent, like most of you said, would be homozygous dominant. But if just one of their children was a non-taster, then we know they are a what? Heterozygote. A heterozygote. Okay, and then we will pick up here. We're going to review research and null hypothesis starting next time. Okay, good job. Review your notes. When do we have a quiz?
Next class.
Next class. When are we probably going to review? Monday at lunch. Good girl. Okay.
Alika, can you come out of it if you can?
No, I didn't.
What guys?